on glycolysis, and it is the second lecture. In the previous lecture, we have discussed about aerobic and anaerobic glycolysis introduction and glucose acquisition and types of glut transporters and some sort of that information. So today we will discuss about the difference between glucokinase and hexokinase and some concepts about glycolysis further. So let's dive into it. First of all, three definitions are very important as far as the glycolysis is concerned. First is the glycogenesis. Glycogenesis means that formation of glycogen. Genesis means that something is being formed. So formation of glycogen, and it is formed from glucose to prevent hyperglycemia. We not need for the blood glucose to shoot, so we need to synthesize glycogen, so it's called glycogenogenesis. The second term is the glycogenolysis. Its lysis means that we are breaking down the stored glycogen that has been formed from the glucose, and we need glucose to prevent hypoglycemia. And the third term is the gluconeogenesis. Something is formed from the scratch. It means that glucose is formed from non-carbohydrate sources. The sources are not carbohydrate related. These are either amino acids or fatty acids. And gluconeogenesis occur in two organs in the body: the liver and kidneys are capable of gluconeogenesis. And in the previous lecture, I have already told you that those organs which are involved in the gluconeogenesis must have GLUT2 transporters. GLUT2 transporters are bidirectional transporters, and these are necessary for the detection of the blood glucose level and for the two-way transport of the glucose. Okay. This is a schematic representation how all these processes are occurring. First of all is the glycolysis. Let me change the color. It's glycolysis, in which the glucose is converted into pyruvate and the pyruvate is converted into acetyl CoA, and then it dives into the citric acid cycle, and a lot of ATP is produced through electron transport chain in mitochondria. Okay, and from pyruvate, glucose can also be produced by a process known as gluconeogenesis. Glycogen can be break down by glycogenolysis and can be formed again by glycogenogenesis. This is just a brief explanation of what's metabolism are being happening in our body. The question arises that why do we need glycolysis? Because there are several other energy metabolism pathways that yields high number of ATPs as compared to glycolysis. Talking about glycolysis in aerobic pathway, it yields 7 ATPs and 2 pyruvate molecules, and anaerobic yields 2 lactate and 2 ATP molecules, but these are way lower than those we obtain from other metabolism pathways. For example, in fatty acid oxidation, the oxidation of palmitic acid and stearic acid yields 106 and 120 ATPs respectively. It means that fatty acid oxidation are yielding way more than glycolysis. So why we need glycolysis? It's a question. We need glycolysis to generate ATP anaerobically because other mechanisms cannot generate ATP without oxygen, and glycolysis is the solely pathway in which ATP can be generated in the process anaerobically. Those cells in the body which cannot function with oxygen, or maybe they don't have mitochondria or they have poor supply of oxygen, must need some sort of pathway to generate their ATPs. Talking about those cells are RBCs, our red blood cells. Despite they carry oxygen, but they do not use it for themselves. RBCs, retinal cells, and corneal cells of our eye do not have mitochondria. They lack mitochondria, as you know that mitochondria undergoes oxidative phosphorylation to generate ATP. So these cells do not have mitochondria. So how they gonna yield ATP aerobically? They do not have any other option to rely on anaerobic mechanisms. Other cells are white muscle fibers. White muscle fibers are those muscle fibers which are like they give instant power and instant energy. For example, you see the birds have like wings, and the muscles which are attached to their wings, they generate a sudden thirst, right? So these are white muscle fibers. These are also known as the fast twitch fibers or type 2 muscle fibers. These are white, but name is suggesting that they does not have myoglobin. What is myoglobin? Myoglobin is the stored form of oxygen, and hemoglobin is the transporting form of oxygen. But they lack myoglobin. It means that they have oxygen shortage. So how they gonna yield ATP via aerobic mechanisms? The third, cells which have poor supply of oxygen is the renal medulla. As you know that in the renal medulla there is the function of concentrating the urine, and renal medulla is supplied by the efferent arterioles of juxtaglomerular nephrons, and through them vasa recta. These are special kind of like arteries or end arteries arises. These are known as vasa recta, and they supply the renal medulla. And we need less amount of blood in the renal medulla because there is the we need for concentration of the urine. We do not need to disturb its hyperosmolality. We need hyperosmolar state there for the concentration of urine. So if there is high blood supply or like sufficient blood supply with oxygen, so the hyperosmolality would be washed away. So we do not need that in the renal medulla. So in the renal medulla cells, they also uses anaerobic mechanisms for them to generate ATP. Okay, the next topic is the preferred fuel. What are the preferred fuel for anaerobic cells and aerobic cells? Think logically that anaerobic cells, as I have discussed, like RBCs, retinal cells, corneal cells, white muscle fibers, and the renal medullary cells, they rely only on glucose for their generation of ATP so that they can perform anaerobic glycolysis. Okay, anaerobic cells only depend on glucose, while aerobic cells, they have sufficient amount of oxygen, so they can perform glycolysis as well, and they can also perform fatty acid oxidation. Among these two, from fatty acid and glucose, which one is the preferable fuel for aerobic cells? It's of course fatty acids, because as I've mentioned above that the fatty acid oxidation yields way higher number of ATPs as compared to the aerobic glycolysis. It only yields 7, but as compared to the fatty acid oxidation, it yields 106 and 120. These are way higher number of ATPs we are obtaining from fatty acids. Aerobic cells, the most of our cells in the body are aerobic. Cardiomyocytes, our skeletal muscles, red muscle fibers, all of them are aerobic cells, with the exception because neurons are very vulnerable to the ischemia and hypoxia. So neurons can use only glucose for their ATP production. It is because that when fatty acids are in the blood, they are conjugated with albumins or plasma proteins in the blood, and they form strong bonds with albumins, and they do not cross the BBB, the blood-brain barrier. So how they gonna be accessible to the neurons? They are not. So neurons also depend only on glucose. There is a clinical situation given here, so let's read it and try to understand it and try to find your own answers while reading it. Okay, a 5-month-old baby failed to respond well to breastfeeding. Okay, she is not responding to breastfeeding. She was switched to cow's milk. okay, at the age of 4 weeks. She was taken to hospital and had symptoms of screaming after eating, but there was no diagnosis. Suspecting it to be lactose intolerance, it means that lactase enzyme is deficient. Her body cannot metabolize lactose. Her parents eliminated milk from her diet, but it did not improve her condition. It's the main point. It was found screaming bouts after initiated after a child drank juice, which cause formation of gas and distended abdomen. No enzymes deficiencies were found, and biopsy of intestinal tissue would indicate which deficiency of the following. It means that lactose intolerance was suspicion, but it is like eliminated from the choices because no enzyme deficiencies is written in the question. It means that lactase is eliminated and sucrase, it's also enzyme, it's also eliminated. GLUT2 transporter and GLUT5 transporter are left. GLUT2 is required for the two-way transport and it is present, as I have mentioned, in the liver and it is present in the kidneys. One of the hereditary conditions is syndrome known as the Fanconi-Bickel syndrome. It is a syndrome which occurs due to the deficiency of GLUT2 transporters in the kidneys in the PCT proximal convoluted tubules. It leads to the like reabsorption disturbances. It means that glucose will be washed away in the urine. The polyuria will occur. Polydipsia, increased thirst will occur, and hypoglycemia because glucose is being washed away by our kidneys, and short stretcher. These are some hallmarks for the Fanconi-Bickel syndrome. But in this child, are any one of these is mentioned? No. It is mentioned that she has formation of gas after drinking juice and distended abdomen. Okay, while in the Fanconi-Bickel syndrome, it's hypoglycemia and polydipsia and polyuria and glucosouria. So these also the GLUT2 is eliminated from the options. We are left with GLUT5. GLUT5 is a fructose transport. As I mentioned in the previous lecture, that GLUT5 is required for the transportation of fructose. It means that after drinking the juice may contain fructose. It may be fruit juice, and fruits are full of fructose. So fructose is not absorbed, and fructose stays in the intestinal lumen. When it is present in the intestinal lumen, it will attract water, and it results in osmotic diarrhea. Also, the bacteria of our colon, large intestine, converts fructose to methane, and methane is a gas which results in bloating and flatulence, distended abdomen, and formation of gas. So like this we are going to like correlate the question hints and the options given in the answer. Formation of gas is linked to the flatulence and bloating and distended abdomen as well. So GLUT5 is the correct answer among these. The difference between hexokinase and glucokinase, what are the difference between these two enzymes? First of all, when we take glucose, when the glucose enter into the cell through GLUT transporters, it can be either 1, 3, 5, not 5 because it's fructose, it can be 7, anything. When glucose enters into the cell, as I have told you that GLUT transporters are two-way transporters, okay, and these are processed by facilitated diffusion. It means that as for when the glucose enter into the cell, its concentration inside increases, and when its concentration inside increases, it may go back towards the extracellular space, because these GLUT transporters are facilitated diffusion transporters. They work by noticing the concentration gradient, so we do not want to lose the acquired glucose. We need to trap that entered glucose. So how we gonna trap that? It's trapped by adding a phosphate to the 6N of the glucose. We will convert glucose to glucose 6 phosphate. And how we gonna do that? This is done by enzymes known as glucokinesis and hexokinases. Glucokinase and hexokinase, these are the most important enzymes because these are involved in trapping the glucose inside the cell. What is the difference between these two enzymes? Okay, just think logically. Okay, glucokinase is present in the liver and hexokinase is present in most of the body cells. What is the function of liver? Liver function is to regulate the amount of glucose in the blood. It means that glucokinase has to perform some functions of storing, right? Liver has to store excess glucose and it has to release when glucose is required. So glucokinase is like a jet, okay? It is like a jet, and hexokinase, which is present in most of the tissues, is like a helicopter. Most of the tissues, for example, we have taken a meal, and after that, a lot amount of glucose is released from the portal circulation and goes into the liver. So, for a large amount of glucose, we need glucokinase for its storage. It means that glucokinase is such an enzyme that work under high level of glucose. It does not work under the low glucose concentrations. It's like a jet. Jet has capacity of 200 to 300 people, but helicopter can accommodate only 10 to 12 people, right? So glucokinase is like a jet. When there is enormous level of glucose in the blood, glucokinase will work. But when there is some amount of glucose, a very low amount of glucose in the blood, so hexokinase will start to work. Okay, it means that hexokinase has high affinity for glucose. Even if there is small amount of glucose, it will gonna pick it. But glucokinase, it requires a lot of glucose for it to work, so it has low affinity of glucose. It is present in the liver. What is Km? Km is known as the Michaelis constant, and it is very important for understanding this functioning of these enzymes. Michaelis constant is defined as the substrate's concentration at which the half of enzyme is saturated. It means that when we are talking about glucokinase, does it require low substrate concentration or does it require high substrate concentration? It's like a jet. It requires a lot of substrate. It requires a lot of people, right? So it has high Km. And hexokinase, it requires low glucose concentration. It requires less number of people. So it has low Km, but it has high affinity, and glucokinase has low affinity for glucose. Okay, as we're discussing that hexokinase becomes saturated at very low blood glucose concentration, it means that whenever the glucose concentration is low, hexokinase is gonna work. It has low Km, it has high affinity. But glucokinase will only work when the blood glucose level is very high. It becomes saturated at high glucose concentration, so it has some high Km and it has low affinity for glucose. Okay, regulation of these enzymes. As we discussed that glucose is converted into glucose 6 phosphate by action of hexokinase and also glucokinase. But when these enzymes know that when to stop and when to be activated, thus the product glucose 6 phosphate. Glucose 6 phosphate is a signal for hexokinase to stop. It means that hexokinase is activated when the blood glucose level is high. It means that when the blood glucose level is high, the cell is taking the glucose and immediately that glucose is being converted to G6P. It means that hexokinase should work in this condition. but when it should stop, it must know when to stop. Glucose 6 phosphate is a negative regulator of hexokinase. It means that when a lot of G6P is being formed, it signals to the hexokinase that stop its activity. A lot of glucose has been converted to G6P, so we do not need your activity now. And glucokinase is also regulated by these mechanisms when blood glucose level is high, but it is regulated by glucokinase regulatory proteins. Okay, and glucokinase regulatory proteins are very important. These are present inside the nucleus of hepatocytes. Why we are talking about hepatocytes? Because glucokinase works in the liver, right? And the liver cells are hepatocytes. Glucokinase regulatory proteins are present inside the nucleus of hepatocytes. When the blood glucose level rises to very high amount, these proteins are formed by the process of central dogma, right? These are formed from the transcription of the DNA to messenger RNA and translation from messenger RNA to proteins, and they are present on the these are present on the surfaces of the cell, and they regulate the glucokinase to work. When the blood glucose level rises, these glucokinase regulatory proteins regulate the glucokinase to convert glucose to G6P. And when glucokinase activity stop, it is stop when G6P converts into the fructose 6 phosphate. Glucose to glucose 6 phosphate, and after that glucose 6 phosphate is converted into fructose 6 phosphate, and this is the regulator which inhibits the glucokinase activity, right? G6P was inhibiting hexokinase while F6P is regulating the glucokinase. This is some sort of graph that represents both actions of both enzymes. There is the performance on the y-axis and glucose concentration on the x-axis. As I've told you that hexokinase has low Km, it means that it became saturated at very low blood glucose concentrations. For example, at this point, it became saturated and it reaches the Vmax. Vmax is the velocity at which it's performing the maximum. And glucokinase has large Km. It means that it requires a lot of substrate to become saturated. Okay, for example, this is the Km for the glucokinase, and then when its Km is reached, it reaches the Vmax and it starts working well. This is the difference between these two enzymes. Let's revise all of that. First of all, glucose enters the cell and it is trapped to glucose 6 phosphate. It's gonna occur by two enzymes, glucokinase and hexokinase. Glucokinase is a enzyme present in the liver cells, and hexokinase is present in the other tissues of the body. Glucokinase has large Km. It means that it works under large amount of blood glucose, and hexokinase works under low blood glucose levels. And glucokinase is regulated by F6P, it means fructose 6 phosphate, and hexokinase is regulated by glucose 6 phosphate. Okay, and one last thing to be mentioned is that conversion of glucose 6 phosphate to fructose 6 phosphate requires an isomerase, because these both have same structural formula. It is known as phosphoglucose isomerase. And then our discussion ends. I will see you in the next video with the